recite the Holy Quran verses?
Muted, ma'am. Now muted. أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. اقرأ باسم ربك الذي خلق. خلق الإنسان من علق. اقرأ وربك الأكرم. الذي علم بالقلم. علم الإنسان ما لم يعلم. صدق الله العظيم. In the name of Allah, the most beneficent, the most merciful. Oh, the Prophet read with the name of God who created the man, who taught by pen, who created the whole universe. And he taught the man whatever he doesn't know. அளவற்ற அருளாளன் நிகரற்ற அன்புடையவன் அல்லாஹ்வின் பெயரால் ஆரம்பிக்கின்றேன். இந்த வசனங்களின் தமிழாக்கம் நபியே இறைவனின்
சார் அன்மியூட் பண்ணிக்கங்க. சார் மியூட் ஆயிருக்கு சார், கொஞ்சம் அன்மியூட் பண்ணிக்கங்க.
சதகல்லாஹுல் அலீம். இறைவன் உண்மை கூறிவிட்டான். மிக்க நன்றி. Thank you.
Holy Quran. Is this audible?
Yes, ma'am.
Okay. So first, the start of the program is welcome address. So greetings to everyone. It's my immense pleasure and duty to welcome the August gathering. At the outset, I pray Almighty to shower His blessings for the grand success of today's international webinar. First, I welcome our Secretary and Correspondent, District Collector, Dr. Anish Shekhar, IAS, who always show his positive vibes for every event. Sir is not with us today because of his busy schedule, but it's my duty to welcome you, sir. I'm indebted to welcome our most kind-hearted principal, Dr. Muhammad Aslam, sir, to deliver his presidential address after the welcome address, and who always hoists the flag of success for the welfare and the betterment of the students, as well as the staff members. He is always ready to initiate innovative ideas and implement various technocratic tools to the staff members, and is a very busy man. I welcome you, sir. Next, I extend my sincere welcome to our chief guest, Dr. S. Senthil Kumar, Assistant Professor of Department of Applied Biotechnology, University of Technology and Applied Science, Sultanate of Oman. He is the most promising and energetic personality who is ready to impart his productive ideas on innovative lecture on impact of diseases in shrimp aquaculture and the diagnostic method. Sir, I welcome you, sir. Next, the presidential address by our great principal and the pillar of the success, Dr. A. Muhammad Aslam, sir. Sir, kindly deliver your presidential address, sir.
Thank you, ma'am.
Thank you, sir.
Respected Head and Associate Professor of Department of Zoology, Dr. K. Samim Rani, distinguished Chief Guest and Resource Person of today's national webinar, Dr. S. Senthil Kumar, Assistant Professor, Department of Applied Biotechnology, University of Technology and Applied Sciences, Sultanate of Oman. And my learned colleagues and my beloved students and other participants from various other institutions. Today is an important day in our college because an international webinar has been organized by Department of Zoology, ably headed by Dr. K. Shamim Rani, and the faculty members, Dr. Mumtaz, Dr. Muhyiddin Abdul Qadir, and Dr. Rajapriya. So I congratulate them for taking efforts, because our college is very busy. For the past 2 weeks, we are submitting the annual quality assurance report for NAC accreditation. So everyone is very much busy, especially Dr. K. Shamim Rani also taking lot of efforts and help in submitting that AQAR. In spite of that busy schedules, he was able to organize a international webinar. So I once again congratulate her. So today's topic is impact of disease in shrimp aquaculture and its diagnostic methods. That will be very useful for the students. So the resource person is going to give you a lot of ideas regarding this topic. So all the participants give you a patient listening to his talk and get most out of his talk. So I wish to congratulate once again the Department of Zoology, and I take this opportunity to thank them for giving me this opportunity to deliver the inaugural address in this international webinar. Thank you.
Thank you, sir, for sparing the valuable time to us. Since I saw you in the chamber, you were so busy. So I concised my welcome address. Okay, thank you, sir.
Thank you.
Sorry for the break in between the welcome address. So I gave the preference to the presidential address by the principal. Any event cannot be happened in overnight. Full efforts have been taken by the faculty members of Department of Zoology, Dr. Mumtaz, Assistant Professor of Zoology, Dr. Abdul Qadir Muhyiddin, Assistant Professor of Zoology, Dr. Rajapriya, Assistant Professor of Zoology, and Professor M. Sikandar Basha, Associate Professor and Head in the Department of Botany. Without their support and hard work, the webinar will not come out fruitfully. From the depth of my heart, I welcome each and every one of this femina as one of my family members. Next, I welcome all the HODs and the other faculty members, technical staff members, office staff members. I welcome one and all. I sincerely welcome the staffs from other colleges, universities, and fraternity professors from abroad, especially from Dubai, Riyadh, and Oman, and students from other colleges. I welcome one and all. So I like to say a few words about the chief guest. He seems to be a simple man, but a highly renowned person in their subject, in their knowledge. 1 minute. Okay. Sir did his undergraduation at the Manonmaniam Sundarnath University and MSc at Gandhigram University and PhD from the Faculty of Science, Marine Biotechnology. His field of specialization is medical microbiology, genomics, PCR. Nowadays, PCR is most important one for diagnosis of COVID. So he's very well versed in PCR, screening of human pathogens using molecular methods, phylogenetics, chemical analysis of water and management of ISO 9001, 14,000 and 22,000. He worked as an assistant professor in Anna University, MGR College, Hosur, CCMB, that is the most renowned scientific center, that is Center for Cell and Molecular Biology, Hyderabad, who worked in pathology lab and at Kingdom of Saudi Arabia. He did his postdoctoral fellowship in the Department of Biotechnology, CCMB, Hyderabad. He received various fellowships, out of that DANIA, Denmark, six-nation collaboration, and highly remarkable. He conducted Cardio Sweden project as a socio-economic fisher folk. He established microbiology lab in various universities in and abroad. He guided MPhil students for the project study. He developed the best one he developed mitochondrial DNA marker. It's a very best concept for the study of corals. He worked in various standard companies in Riyadh and abroad. His achievements innumerable. He participated in more than 50 international conferences and seminars and published more than 50 research papers in international reputed journals. He received many research funds from Research Council. He was invited as a resource person in national and international arena. Such a big profile, but with many laurels and credits, he possessed simple and kind enough to accept our invitation immediately as a resource person, even with a busy schedule. I welcome you, sir, wholeheartedly.
Thank you, ma'am. Thank you so much.
And one thing I should not forget, that is, I must pronounce my gratitude and wishes to my ever-loving student. Now he's working in a very big concern, Mr. Sharavanan and Mr. Fahad from Dubai, who is the keen success of this function. I invite the eminent speaker, Dr.Senthil Kumar sir, to deliver his valuable address. Once again, from the depth of my heart, I welcome you, sir. I welcome you, sir, on the part of my college and my department. Thank you.
Thank you so much, ma'am, for your nice introduction.
Thank you.
And all the faculty members and all the students, research scholars, and other faculty members from various institutions. Thank you once again for giving me the opportunity to share my experience on stream disease impact on stream disease, impact on diseases in stream aquaculture and its diagnostic methods, which I have worked in past almost 7-8 years. I worked on this field as a pathologist, stream pathologist. Mainly, I have developed on molecular diagnostics methods. So I just want to share my experience with you, all of you, today. Thank you so much. Let me share my screen. I think you can able to see my screen, right?
Yes, sir.
Okay. Everything's fine, both audio and video.
Yes, sir.
Thank you. So let me start. As the title itself is a self-explanatory, that the impact of diseases. So that aquaculture sectors in South Asia, especially, we are struggling with a lot of unknown diseases like bacterial or viral or parasitic or microbial infections. So it's a big challenge in front of us to address this issue, to come out to be a successful aquaculture sectors. So I just to brief you that what is the most significant part in the diseases especially in shrimp aquaculture. So the issues and challenges in shrimp aquaculture in Asia, as I told you, the predominant is the diseases. So we are always like almost 100%. We are struggling with the diseases, the management issues. So how we can tackle, or before going to the actual topic, we need to know like what are the diseases, common diseases in aquaculture, especially in the shrimp population. So according to that, we will proceed that. Okay. So this is the challenges, as I told you, the top issues and challenges is diseases, because it is not only affecting the what you call the population, and the huge impact on the market prices and the production cost, how much we are invested. So whatever we are investing, we have to take it back. So that is the ultimate aim of the aquaculture sector. So which is not an easy job, because we are always handling the live animals, which could happen whatever happen, it's not it's our control. So though we have to be aware of the common diseases and its impact on the issues. Okay. So let me start with the anatomy of the shrimp. So you might have studied in your curriculum, maybe. So I just want to show you the first picture. The slide is itself like the anatomy of the shrimp, because we need to take as different body parts for the sensitivity of the diseases which you are going to diagnose, especially for the monodon baculovirus. It is MBV or HPV. It will be in the hepatopancreas region, where you can find in the near the carapace. Okay, and this uropods and the pleopods or the gills are very sensitive portions for the Wssv or IHNV, TSV, lots. I will just brief you what are the diseases according to the Office of International Epizoonotics, that is called OIE. So these are the things you need to remember, that why we put this anatomy of shrimp in the first slide. So the value of the test, as I told you, it is characterized by its sensitivity and specificity. So each techniques, whatever we are going to adopt in this disease diagnostic procedure, has different purposes of use. So from the practical point of view, they can be classified as field techniques and laboratory techniques, because now you know that aware of that what are the field techniques or what are the antigen-based detection which we are using in the airport, like for the detection, fast detection for the COVID-19. So it can be classified. So field techniques, like it can be done in situ and with a little equipments, and the results are available within a short period of time. Whereas in the laboratory techniques, it requires the support of a more sophisticated laboratory, then results may take a longer time. So in order to minimize the what you call the time taken for the diagnostics, we need to be very conscious, like which techniques you want to adopt for what kind of disease you are come across. Okay, so these are the classification from the practical point of view. So before any diagnostic procedure, especially for the disease diagnostics, the sampling size, in order to attain the 2% of prevalence, that is the most important thing as the international standard. We need to have, according to the population size, how much individuals you have taken for the sampling in order to achieve the 2% prevalence, and make sure that you are not miss any information. So that is the main point. So these are the, according to your population size, you need to take a minimum number of sampling. So if you have a more than 1 lakh population, if in a culture system, so at least 150 individuals you need to be sampled for the diagnostic procedures for the disease, especially for the PCR. Okay, so these are, as I told you, these are the OIE listed crustacean diseases so far. It may be increasing, like there are a lot of unknown diseases it causes, and the cause or the causative agent, we are still they are working on it. It's not a end. There is no end for this particular diagnostics or procedures. Okay, so these are the common so far which I have worked on this various diseases of bacteria virus. So these are the main diseases: Toro syndrome, TSV, white spot diseases, which is you are most commonly you might have heard this white spot syndrome or white spot diseases, and yellowhead virus, and tetrahedral baculovirus, perical baculovirus, baculoviral midcut gland necrosis, infectious hypothermal hemopoietic necrosis, infectious myonecrosis, necrotizing hepatopancreatic, infection of hepatopancreatic parovirus, infection by Mullerian virus. So these are the listed OIE listed virus. So we will just, I will just brief you that what could be done, what are the symptoms, external symptoms, and what are the histological observations done so far. Okay, so these are DNA viruses, as I told you. These are a brief introduction about the structure and the family belongs to the virus. So white spot syndrome virus, we call it as a Wssv. It's a Wssb virus. It's a Pneumoviridae. It's a double-strand DNA. It's a size approximately 120 into 270 nanometer. So it is self-explanatory, the slide. So you can have an imagination that how much of the size and which belongs to it's a DNA virus or RNA virus. So white spot, monodon, baculovirus, baculoviral, mid-cut gland necrosis, these are belongs to DNA-based virus. So till it is continued, IHNV, HPV, lymphoid organs, spheroids, these are belongs to DNA-based. So there are RNA-based virus. They have classified DNA-based and RNA-based. So yellowhead virus, gill-associated, and rhabdovirus, lymphoid organ vacculation virus, Toro syndrome. These are belongs to RNA dependent. So we have classification that DNA dependent and RNA dependent viruses. So these are the susceptibility clinical diseases by species. So the pinase monodon is the most tastiest species as well as the most susceptible in which all the what you call DNA dependent virus infection. Seleucus venomi also, it is now pinase indicus, venomi, Styloristus, Japanicus. These are belongs to the disease susceptibility, that especially pinase indicus. Now they are widely cultivating in Asian countries, and here also there are Oman also there are 2 companies they are working on this pinase indicus culture. And here the advantage of is the what you call the water. The water quality is highly impact on this, especially in the disease carriers. So here it is as of now we are not come across in any disease prevalence so far. So why I'm telling that is essential for the what you call the quality of water quality. So relevance of main virus based on the economic impact. So DNA virus, as I told you, the Wssb is the most dangerous virus. Once this virus infected, the entire system will be collapsed within 3 days. So it's a most sensitive. That is why it is a most familiar also, the white spot syndrome virus. And RNA virus like yellowhead virus and gill-associated virus and Toro syndrome, these also have impact same like Wssb. These are the stages of disease expression, like acute, like it invariably all stages it will infect. Like example, it's starting from egg, juvenile, adult. There is no criteria that it will affect only the juvenile or adult. So invariably all the stages of the what you call the life cycle of pinnae species, it will have impact. It will affect. So it is acute high mortality, as I told you, this Wssv, even that IHNV also, and it will be slow growth, that is the impact. And HPV also, it is a slow growth. So these are the impact, huge impact, because we have to attain the maximum size and whatever we are invested. So that cannot be possible if the infections, once you come across in the infection. So these are the different agent, like different diseases, and which stage it will affect and what are the impact of these particular diseases. So like MBV and baculovirus and BMNV, this is affect invariably all stages of life cycle, and the impact. It is most of the mortality. It leads to be sure mortality, and also it is slow growth. So we can have regular monitoring that, okay, this according to your feed conversion, like FCR, you know very well how much of feed you are given, and the growth of the individuals. Based on that, you can convert the FCR ratio. So if it is slow growth, if you are noticing that some sort of infections are going on, so you have to be aware of that, what kind of agent is infected. So these are the RNA-based virus. What are the different stages of the disease expression, like yellowhead virus, gill-associated virus, RPS, LOV, Rio virus, Toro syndrome virus. So all stages almost, and till the impact, they are confirming it. Some of the unknown diseases or some of the diseases still work is going on. So what are the diagnostics method? As I told you that the yellowhead virus and white spot syndrome virus, these are the most impact or huge impact, high mortality you can come across. So these are the different protocol, different diagnostic procedures like external signs and what are the rapid techniques are available, what are the histological procedures we need to be followed for the disease confirmation, and the electron microscopy. It is, if possible, you can go for that, and DNA-based techniques, various techniques. So what the selection of, as I told you, the selection of diagnostics method also, which depend on the purpose which you are going to do. So if in case of a disease outbreak, you need to go for all kind of steps, or if you are going for just for deduction of the agent. For example, you are cultivating, you are culturing some, you are doing some culture, and you want to export your product in order to get your certification that your product is free from all these listed viruses. That is for screening purpose, only for certification issue, that can be possible. So according to the purpose of which you are doing, so that will be the what you call the selection procedures also, it depends. And what are the disease outbreak, like if it is external signs like unspecific, they are indication of an anomaly, it cannot be used for diagnostic purpose for this in case of disease outbreak, because we are not aware of that unknown causes or unknown diseases or unspecific something happen. So that is a disease outbreak. So you have to follow, and what are the OA listed protocol we need to be followed. So these are the external signs. I just want to brief you that what is the yellowhead virus. You can see that if it is infected, that it is become a pale yellowish in the cephalothorax region due to the pale and swollen hepatopancreas. And as I told you, this is the hepatopancreas region. So where you can able to see the differences of the healthy and diseased individuals. So this is the external signs which we have to be aware of that. And then if you open it, you can see that this completely disintegration of the hepatopancreas as well as yellowish color also. So this indicate that this particular individual has infected yellowhead virus. So what are the external signs of Wssb? There is a white spot inclusion in the sides of the cuticle where you cannot remove by scratching this white spot. It may be sometime it is associated to the water hardness also, but if it is associated with the water hardness by scratching, these are the what you call the to confirmation that it is white spot, or it is really infected by the virus, or it may be cause of the other environmental factors. So that can be possible to differentiate. You can just scratch by the cuticle of the white spot. You cannot remove this white spot in case of infected by white spot syndrome virus. So whereas if it is associated with the other environmental factors, it can be removable. So these are the external signs of white spot syndrome virus, the white spot. So what are the rapid techniques? I just focusing on the Wssb and YHV, yellowhead virus. So there are a lot, but with this 1 hour of period, so at least you should know what are the things are available for the diagnostics. So what are the rapid techniques? It can be performed in the form where you are doing actual operations. The results can be read in 1 or 2 hours, minimum hours, and low sensitivity. Sometimes it leads to low sensitivity in case of clinical cases. And the positive results, we need to confirm the disease. The negative results, it does not confirm the absence of the disease. And there are guilt staining is available. Hemolymph smear also we can able to do for rapid techniques. So guilt staining, you can just, it's allowed to identify yellowhead virus, white spot syndrome, and infectious IHNV, and vibriosis, mycosis, and epipyones. So these, what you need to do, remove the gills, fix it with the Davidson HCl, and 50% of HCl, replace the glacial acidic with the 50% hydrochloric acid for 1 hour, and stain with the hematoxylin eosin. It's a normal staining stain, and then dehydrate with ethanol and keep in xylol till it's mounted, and then you can observe, you can able to see that the disintegration of nucleus of various cells in the gills, you can be able to observe. And there are some powdery inclusions also, some of the nucleus where the complete disintegration also you can able to observe. So in such cases that, okay, this is some sort of infection, but we need to confirm by PCR. So these are the histological photograph of the gill staining. So you can able to differentiate that the disintegration of inclusion of all the bodies, all the nucleus. So this can be the first, what you call the rapid techniques which you can observe in the field. So the next, the hemolymph smear. So, just to draw the blood from the, from the individual, it shows the morphology of hemocytes and the presence of bacteria and parasites. There are two types of morphological changes you can able to observe. Pycnosis, it is a condensed and reduced nucleus or basophilic, or karyonexis, it's a nuclear fragmentation or intracytoplasmic bodies you can able to observe in the hemolymph sphere. Collect the hemolymph, as I told you, in a syringe with a Davidson water, just to replace the glacial acidic acid with distilled water. The Davidson fixative, it's a common protocol. You can able to get, even if you Google it, you can able to get, and lot of research papers also available for the Davidson fixative, which you can use for histology of this. So there is not much expertise on the histology, as you know that. In southern part, we are able to struggle for every techniques also. So that can be, if you are interested in pathology, you can develop, like you can just collect some samples from the market, and you can do try it your own. If you have the facilities. So this is what the way you can practice and you can expertise yourself. So smear it on the slides and stain with the H&E, and you can able to see that you see that normal and abnormal, the hemocytes of the individual. So other field techniques also available. As I told you, it's a non-destructive method. By scratching the cuticle, you can able to identify what are the fouling organisms. As you know that it is very, it's a very, inevitable that the carapace of the crustaceans, you will find the fouling organisms. So that also have the impact, because it's like a parasites. So it will also have impact on the aesthetic appearance of the species. If it reduce the market value, as well as it causes various associated problems also. So that can be also identified by scratching the cuticle. You can able to find out the fouling organism, and the fresh mound gills or fouling mycosis or diatoms, which are attached on the surface of the carapace or the shells of the the shell of the shrimp. And then feces, you can collect the feces, fecal pellet, and you can add a drop of malachite green, and you can observe whether it is infected monodon baculovirus, or the even the appendices, it's a fouling mycosis or Taurus syndrome also. That can be possible with other field techniques. So these are the non-destructive and destructive method. They classified the two different field techniques. So destructive method, the scratching of the midcut, you can able to see the gregarians, the dissection of the lymphoid organ. You can able to see the spheroids, the smear, like a hepatopancreas, HPV, Mycobacteria, rickettsia, necrotizing hepatopancreas, NHP bacteria, the muscles microsporidae, and bacteria, mycobacteria. So all can be identified this particular techniques. So next is histology. As I told you, this is the confirmation test. So it is a very, it's a highly time taking, and you need to more skill, and you need to identify what is abnormal and what are the cellular level changes in the healthy and diseased animals. So that can be possible by expertise yourself. Like you need to read a lot of slides and a lot of the dissection, lot to be practiced, because histology, it's not easy job. It need to be more practice. So histology, as I told you, it's a global view of the health status of the animal, and the degree of lysine. So it is a slow process. You need to wait for 42 hours in the fixative, and it takes more than 72 hours to get the result. So samples, you need to have a live animals. You cannot do it in the dead animal. So when you have the suspect that some disease occurrence or disease outbreak, so in that case, you need to have some individuals need to be fixed with a Davidson fixative, and you need to do the processing of tissue sectioning. So the presence of what are the main observation of histopathology of yellowhead virus, the presence of pectonic nuclei and karyonecrosis hexes in hemocytes or lymphoid organ spheroids you can able to find, and hemopoietic tissue. So what are the differential diagnostic like TSV and systemic vibriosis also can be possible. So vibriosis is the main cause. There are a lot of Vibrio species which leads to the infection of the viral. So the accumulation of vibriosis like Vibrio horvi, Parahemalticus, these are the main causes. Vibrio horvi, these are the main causes of Vibrio species which causes the infection in shrimp population. So these are the what you call, you can able to see by this photograph set a lot of disintegration of the nucleus, and there are a lot of cellular level changes in case of the disease outbreak or the disease lysins, right? So this can be possible. You can compare with the healthy and diseased animal also. So these are some of the photograph of the histology. So the histopathology of white spot syndrome, like a basophilic intranuclear inclusions, mainly in epithelial tissues, gills and connective tissue. In the first stage of infection of a cell, you can able to see the Cowdery inclusion body, and it can be used for differential diagnosis. So you can see this, as I told you, there is inclusion of, see. So this is what you can able to differentiate the abnormalities of the cellular level changes by practicing yourself. So these are some of the photographs. These are the cuticle. You see, completely it is vanished, all the internal organs and densely basophilic inclusions. And this is the TSV inclusion. This is a normal epithelium, how it looks. So these are the differences you can able to observe. So next is, as I told you, electron microscopy. It's not possible for us, but it is a research purpose. They are now using it. Like you can directly observe the morphology of the virus. As based on that, you can able to what you call identify what kind of viruses are infected based on the size. So it's not used for us. It's not used for routine diagnosis, but it is applicable for the research purpose. In case of an outbreak, it's used for the confirmation of etiology of a new diseases. So these are the photograph of the electron microscope where you can able to see the structure of the virus. See, you can able to see the structure of the viruses. You see, these are the virus. So what are other rapid techniques? So we have the rapid techniques, histology, electron microscopy. So it can be used for cellular level evidence of lysins and changes caused by the pathogens. So what are the antibody and DNA based techniques? And you can go for molecular level detection of the presence of the pathogen, but no information on the possible lysins. The advantages is a very sensitive, quick and specific, and less training of course needed, not like a histology. Histology you can be able to, once you are expert only, you can able to differentiate the cellular level changes. But in case of what you call molecular diagnostics, you need to do the DNA isolation if you are going to diagnose for the DNA dependent virus, or in case of RNA, you need to go for RNA dependent virus. So these are the two differences you need to do. And it is highly sensitive, as I told you, of course, and it is quick. Like you need to wait for only the reactions time. So then you can run the gel. Now there is no need to run the gel. There is advanced techniques also there, the thermocycler. You can able to see the amplification and when the reaction is running. So that can be possible. So as I told you, it depends on the purpose of the diagnosis. So in case of disease outbreak and the detection of the agents, so that can be very much, you need to be aware of that, what kind of. So in case of detection only for certification or screening, the external science will not be present, and field techniques, it's not sensitive enough. The histology also not sensitive because it takes time, and electron microscopy inadequate, but DNA techniques compulsory, it's required. Now, that's why if even the domestic, even your past years, we are more experienced that what is the use of RT-PCR, right? So because it's a RNA dependent virus, they have to do for reverse transcriptase to convert into RNA into cDNA and then go for a specific marker for the particular virus, all right? So this is very much now the recent past. Everyone knows, that even the common layman knows that RT-PCR, right? They don't know what is PCR, but they can pronounce that we have to carry the RT-PCR test report to pass even for the international or domestic, right? So these are the most common what you call sensitive techniques which are the past years we are experienced. So this very much required in for the detection purpose for the disease diagnosis, since there is no what you call cell line has been maintained for the shrimp pathogen. So it is must now they are going for DNA based techniques. So DNA is, you know, that where it is very intact, right? It is not easy to get the DNA out from the tissue, right? So these are things, basic things you already know, that you are already doing a lot of your curriculum, right? So these are the various techniques which is available on DNA-based dot plot, in situ hybridization, PCR or RT-PCR detection, nested PCR, and in situ PCR detection and amplification of the tissue section. So these are the different DNA-based techniques are available, which I got more on this aspect. So, you know, what is PCR? It is a method to detect and amplify a known fragment of DNA. What is a known fragment of DNA? The gene which you are targeting to identify the particular virus or bacteria, right? So this is the known fragment, because in the entire genome, we don't know the genome size. And if you are going for a specific gene, you know the size of a gene. So this is a known fragment of DNA you are going to amplify in order to identify the infection agent, right? So what are the different types of PCR we need to have based on the sensitivity? There is a one step, there is a nested, and a real time. So real time, now they have developed real time also, because a lot of industries are equipped with a real time PCR. Real time PCR, you can able to see the what is going on inside in each and every step of every cycle in the reaction. So what are the other types? There is a multiplex. Multiplex in the sense you are targeting a several DNA in a single tube. So that is called multiplex PCR, because more than one gene you are detecting in a single tube. That means it is RT-PCR, reverse transcriptase PCR, for in case of RNA-based virus. So every PCR is different, every protocol, every kit also is a different sensitivity, specificity, and robustness based on the kit, based on the what you call the method, the PCR method. So what are the advantage of PCR? As I told you, it is a excuse site sensitivity and specificity. Why we are jumping it, especially the shrimp culture industries in this technology, as I told you, there is no cell line to support the growth of these shrimp viruses. So everyone depends on the PCR-based diagnostics in the shrimp especially. So what is PCR intended for? Detecting the advantage of the PCR, it will detect the low amount of virus. So the copy number, even low also, it can be able to detect. That is why it is certified by ICMR or WHO. We need to carry the RT-PCR certificate, right? So why I'm telling, this is a sensitivity, and also it has a specificity, more advantages, even if the low number of the viral copy number also it can be able to detect. So it is based, coming to this, it is used for screening of brood stock and offspring. It's not appropriate for disease outbreak diagnosis. In cases, we need to use the histology. If in case of disease outbreak, like you need to confirm by histology, right? So that is the latest stage. So histology, if you want to really go for histological observations, we need to wait for the infections. So infestations of the particular virus, it will take more time. Like that's why we have a quarantine time, right? So the quarantine time is nothing but the infestation of the particular virus into the host cell. What are the changes are happening, right? So this is what this in the PCR advantages of the PCR. So you know everyone that we have the three principles of PCR, like initial denaturation of the particular virus, because DNA is a double strand. It needs to convert it into single strand, and then it will go for specific the primer which you are targeting for the different kind of gene which you are going to amplify. It will go and bind wherever the complementary sequence found in the genome, and then the polymerase of the tag DNA polymerase will polymerize, and it will extend for the further multiple copies of the known fragment of DNA. So this is what the DNA, the PCR, right? It's a polymerase chain reaction. It's nothing but a thermal cycler. At a particular temperature, it will hold that 95, it is heating up 95 and 54 and 72. So these three different temperature, it is going on, the cycle is going on, denaturation, primer annealing, and the extensions. These are the basic principles of PCR. Everyone knows that already, right? By theoretical point of view. So we need to understand what is PCR, right? So what viruses we can test for with the PCR, it depend on the purpose of the testing. As I told you, screening for domestication program or specific pathogen-free status, or many viral agents as they have been described. The screening for stocking the ponds. So that is only for a Wssb is fine, because they are they cannot afford a lot of money, right? It is not easy. How much you are paying for RT-PCR test? Because in the beginning of the pandemic, we are paying more money, right? Now everyone has developed the facilities, and the government has involved for fixing the rate for everyone. Common man has to pay, right? So that is what the what you call the cost effective also we need to be think of in case of screening. So the screening, Wssb is the most, as I told you, most sensitivity, and it leads to a lot of mortality and loss of impact. So that is why they have approved Wssb for screening of the brood stock or stocking the ponds before the individual introduced into the ponds. Okay. So as I told you, the sample size, it's 150 animals. If it is too much DNA, so it requires the 5 amplification. So it needs to dilution. So if you dilute, you will get a loss of information. So the smaller size, if it is a post larvae, that is a 60 individual post larvae, you have to sample it. If it is 10% prevalence, you have to 30. So dilution, if you are diluting the DNA concentration, because for amplification of in the PCR, there is 20 nanogram of DNA is fine. It can able to amplify. But when you are diluting it, when you are taking 150 individuals of animals to sampling, you will get too much of DNA, right? When you are isolating the DNA, the concentration of DNA will be high. So if you are diluting it, you will lose the information. So we need to be practical and what you call it. We need to be very careful. We should not lose the informations, as well as at the same time, the cost-effective management also you need to be cost-effective. Also, you need to be concerned, right? So that is, you need to be aware of that. You need to be aware. Okay. So sample tissue, what are the target tissues of the virus? As I told you, some tissue present the risk of infection. Some maybe give a false result. So post larvae 10 onwards, you need to have the eye stalk, and that is why I put in the anatomy of the shrimp, right? So now you need to understand what is the correlation. So hepatopancreas or feces or egg or nauple. So some of the tissues will give a risk of, because there is a lot of PCR inhibitors, as I told you, because it has, it's not easy, right? And it is most, what you call cost also, we are involving more cost because the enzymes are too costly. At the same time, what you call, there is an inhibitor. So if you fail one reaction, you will be losing some money. So that all need to be considered when you are working on the PCR-based diagnostics method. So what is the bestest sample? Broodstock after spawning, offspring, weaker animals or formalin. Even now it is preserved in formalin. We are able to, there is a protocol available to get a DNA out of the formalin preserved animals also. And the post larvae before stocking better than the egg carnoplia. You need to have a rinsing of the specimens before packing for the laboratory for no EDTA or malachite green, because this is the potential inhibitors of the PCR. So you need to be aware of these things also. Why can have be false results? The false positive may be a contamination. It can be environment or equipment or sample or reagents. So false negative may be some of the inhibitors of the PCR, the volume of sampling which you are taking, maybe it's a human error, inadequate extraction of nucleic acid. If you are not thorough or if you are not sure about the methodology of DNA extraction, it is end up with negative results. So equipment failure, reagents problem, poor specimen quality, these all need to be considered for the PCR based diagnostics method. Okay, how to identify when the results are positive? By using the appropriate control. We need to have a big, especially for the disease diagnosis, because we are not going for sequencing. When you go for sequencing, at least you need to know, you will know that, okay, this is belongs to Wssb or TSV, but we are just stopping with the PCR with electrophoresis. So you must be very careful, and you must be what you call make sure that you are giving 100% result, authentic result. So we need to run the positive control, negative control, environmental control, internal control, sensitivity control. These are mandatory for PCR-based diagnostics methods in the disease especially. So you need to be consider all these standards, all the controls once you are running with your reagents, once you are running with your sample, these all need to be taken care. So what is the internal control? It clears the possibility of the false negative results. The sample needs to be repeated after further purification or not. So that will give us the indication of different controls. So what are the controls that should be run with every batch of sample? Positive and negative. So you need to know what is the positive control, what is the negative control. Positive control will supply with a kit if you are using kit based. If you are not using kit, you can if you are going for conventional procedure, we need to be aware of, we need to be make sure that a positive control is running, like a known infected animals already it is there. If you have a Wssb DNA, then you can run along with your sample so that you can make sure that you are run all the controls to give appropriate results. So environmental control and sensitivity control. So these are to be monitored regularly so that your lab is equipped, or your lab is, or what you call give you authentic results. So that can be confirmed with the different controls. So other than that, there is an in situ hybridization. It's mostly a research tool of pathogenesis studies. It's not more sensitive than histology, but it will help us for the light infections also for in situ hybridization. So these are the what you call the gel picture after running a PCR. You can able to see the gel electrophoresis. You can able to interpret your results. So as I told you, there is a positive. These are the sample which you are running with sample with a severe infection of Wssb, moderate infection, and light infection. So here it is a very light infection. So you can now see that the lane 3 is you are seeing that a single band, and you are saying it is a light infection. In the lane 4, there is 2 bands. You are seeing very light infection. You might have raised a question. Why? Because it is very light infection. How we are saying that it is the 2 bands indicating that it is shrimp DNA. It's amplified. This is what I told you. Multiplex PCR. Multiplex. You are targeting a more than 1 gene in a single tube. So it is a housekeeping gene of the shrimp, and this is your if the infection is more, you cannot get an amplification of your housekeeping gene. So that is all the viral copy numbers which inhibit the housekeeping gene amplification. So that is what the difference. So three different bands. It is a severe infection, moderate infection, light infection, very light infection. Okay, and negative, there should be a one single band, that is why your housekeeping gene is amplified. That means there is no viral DNA is amplified. And 6 is a negative control, you should not get anything. If you are getting a negative band in the negative control, then your PCR is invalid. So that can be, you have to be make sure that. And these are the positive standards which you will give the kit, like a different, like it's a 2000 copy numbers, and this is a 200 and this is 20. So these are the molecular markers of various sizes to identify your amplification of the particular gene. So these are the things must be there in the gel picture. Without running this, if you are producing your results, it's invalid. Okay, so they will not accept even the certification. If you are applying for, you are claiming that your product is free from all these pathogens, and you are attaching the PCR certificate, only the PCR certificate, it is mandatory to run all these samples, all the controls. Okay, so that is I try to stress you. So these are the different, what you call, to show you the real PCR which we have done in my experience. So you can see this every time we have to run all these things. So these are the standard WSSV standard, and these are the negative of your samples of the 4, 5, 6, 7. You can able to see the interpretation of the results. So this is what you need to interpret, and you need to have what you call perfect pipetting. So it is all a very small amount. We are not using for 10 liter or 12 liter or liters based. It is a microliter. So it is a nano, right? It's a molecule based. So everything will be 1 microliter, 0.5 microliter, 2 microliter, 1. So the total reaction of your total volume of reaction is 20, right? So you need to be very careful. So these are the, by practice only you can be expertise for the low volume of pipetting. Now there is a liquid handling system is available, but we cannot afford our every equipments, right? So we need to be practiced more enough on the low volume pipetting, right? So these are about the viral diseases so far I have described. There is a bacterial diseases also. Just to brief you, what are the bacterial diseases which we have come across in the disease, in the shrimp disease especially. There are several genes, genus associated to shrimp. Some are pathogenic, some are epicommensals, or some are beneficial. In most cases, the bacterial problems in growth can be prevented. It may be often it is stress to the animal. So as I told you, once the bacterial infection, that maybe leads to a lot of other infections. So that is, you need to be aware of that. And the larvae are more susceptible for the bacterial infection. You are like you may have experience, like if you are studying zoology as a major, we need to have all the, what you call, like a culture based. When you're handling live animals, it's a different experience, right? So you need to be aware of that, what stage it is susceptible, and what need to be, precautions need to be taken care. So these are the bacterial agents, fouling or different bacterial fouling, vibriosis, mycobacteria, rickettsia, and mycoplasma. These are the different genus which involving the causes of bacterial diseases. And the fouling is leucothorax. It's a fouling organism. You can able to see the histology where the fouling organisms, you can able to see it's a microalgae. And vibriosis, it's as I told you, it is described as a pathogenic. It is also described as a part of their normal microflora. These are the different genus of Vibrios, Vibrio harvey, Vibrio alginolaticus, Vibrio parahemolyticus, Vibrio vulnificus. It's a true pathogenicity in controversial. It is caused a T-brown gill syndrome, and it is infections of the Vibrio bacterium by lysogenic bacteriophage that introduced into the production of toxin. So these are various studies already there for the bacterial infections. So, so these are different forms of vibriosis. You can able to see it's a septic hepatopancreas, and you see completely all the internal parts are vanished in the mollusk, in the shrimp. So this is the indication of a vibrios infection. There is a black spot also on that carapace. And then this is a mycobacterium. It's often described in adult with the lessons and difficult to mold. It's a low infectivity, the risk of zoonosis. The rickettsia, it's an intracellular gram-negative bacteria. It can cause high mortality also, necrotizing hepatopancreatic or Texas pond mortality syndrome. Mycoplasma also, there is a lot of symptoms or associated with the bacterial infection. So this is a Spiroplasma pini. This is the parasitic diseases identified by Leiter. These are the University of Arizona. They are the experts of shrimp pathology. You can search, Google it. There is a lot of literature available pertaining to the disease of shrimp diseases. So these are the fungal disease. So there is a no, as of now, it's a very few people are expertise in the fungal disease of shrimp. So it is a potential opportunity as a young researchers. You can concentrate on the fungal diseases of shrimp. So you can get more opportunities. It's a primary pathogens because it's very difficult to handle fungi. It's right. It's some of these spore producing fungi. It's very difficult to handle it, right? So there are two groups of fungi which is involving in the infections like Mycota and Ascomycota. It is affecting the larval stage and the adult stage. There is two groups of genus fungi which can cause the infections in the shrimp. Larval mycosis, it's a Legnidium calinatectis or Ciroliptinium species. It may cause high mortality, and it is all pinnate stages of species are equally susceptible for this larval mycosis. There is adult mycosis. It is caused by Fusarium solanae. It's a common fungi. It's found in the soil also or in the wooden pieces also. It is also it's opportunistic, and it causes severe inflammation that is usually strongly melanized. That the pigmentations or melanization means decolorization completely. It will affect the aesthetic look. So when you're going to buy a seafood, you can able to see how looks, right? So this is one of the customer satisfaction. So it is affect the aesthetic look of the particular species. So you must be very careful. These are the variable species which are susceptible for the adult mycosis, like Rapanicus, Californiasis are very high. So what are the other parasitic diseases which we have the protozoa, fouling protozoa, Microsporidae, Haplospora, gregarians, and metazoa like nematodes, cestoda, and trematodes. Like human, there is a lot of parasites also which infect the what you call in shrimp. So these are the fouling protozoa genus Zoothamium, and it is the may impair of breathing or feeding movement and molting stage. You can able to see there is a presence of indicating the poor water quality. So water quality is a major role. So when you are doing aquaculture, you should be very careful what are the water quality parameters. You know very well the nutrients like nitrate, NO2, NO3, ammonia, right? Like these are the water nutrients as apart from the pH, POD, COD, whatever. So all the water quality parameters plays a major role for disease management. So these are you know very well. So you must be very careful for the water quality parameters. So these are the populations of bacteria and the algae that horizontal transmit affects all stages of the pinnae. So these are the different parasites which already they have described for the in shrimp, Zoothamium, Anopheles. So these are the different genus of the parasites. So in what is invasive protozoa, that is a Microsporidae, it causes the causative agent of the cotton shrimp. The muscles looks like opaque and the whitish due to the masses of the spores. It requires the presence of carnivorous fish to close the cycle. There are three species which are involving in the invasive Microsporidae. It is affect all stages of pinnae. So you see this, if you remove the carapace, you can see that it looks like a cotton pluck, right? So these are the Microsporidae. You can able to see the Giemsa stain. You see, you can only the disintegration completely disintegrated all the tissues, and you can able to see some black lines. So this is indicating the Microsporidae infection. So other than that, there is a gregarians. There are three species which have been described for the infection. So it is the final coast, it's the intermediate coast or bivalve mollusk and polychaetes. It's responsible for the poor growth. And so it affects all stages of the pinnace indicus, pinnace species. So next is gregarians. As I told you, it's a fresh mount also it is possible, whether it is this particular parasites are infected. So these are the fresh mount. You can able to see it's a hepatoxylin H&E in staining. You can able to see. So the general disease solution, it's we need to improve the biosecurity, like a quarantine, pathogen-free and resistant shrimp strains. It improve the water quality, improve water management and pond design and operation. Use mangroves to filter water, crop rotation to improve the water and sediment quality and remove the vectors. And polyculture, you know, it's very famous in the now Western countries. They are multiple species or they are culturing at a time, and it can be prevent the possibilities of all the infections. So with the fish, you can use shrimp with the fish, crab, bivalves, and other invertebrates and seaweeds. So this is possible for the solution of the disease management also. So what I'm currently doing, we have running 3 different projects, and 2 already is over. The 2 is running. So I have this all is funded by the Research Council of Oman, because we are just establishing like genomics lab, where I'm going to work on the different, because you know corals are very potential indicators for the pollution indicator. So we are going to use differential gene expression using the NGS. The most advanced techniques are next generation sequencing. They can be able to target a multiple gene at a time, like a transcriptome genomics, genome sequencing. We are going to identify what are the stress which causes the mortality of the coral. So this is for the conservation point of view. You know that coral ecosystem, how much important to the marine ecosystem, right? So it's a most productive ecosystem, and it is the most what you call highly conservation point of view. We need to highly you need to take a more effort on to conserve this particular ecosystem for the future generation, right? So with that, I'm just finishing my talk. Thank you for your attention. If you have any questions, you are very welcome to ask me. And thanks once again for inviting me for and giving me opportunities. And I thank our one and all present today. Thank you so much, Doctor. So it's open for discussion.
For your awesome presentation.
Thank you, ma'am.
And it's highly skilled technical applications in dealing with the histopathological studies. Really great. Looking at one of the senior most scientists and eminent personality in histopathological studies in shrimps. I'm so thankful for being with us, sir. And my students received certain information from you, from the light talk. Totally, it's based on research applications, but she learned many things, how to handle the pipette and the instruments. Okay. I'm so thankful. I'm so thankful, and no words to find same gratitude. So thanks of you, sir.
It's a very great pleasure, ma'am, because especially for the south side, we are not much aware of what is going on. And it is very difficult to come out from the place. So, you know that when I got my postdoc opportunities in CCMB, there are very few students from the south side. So we need to come out and we need to explore the whole world. So that is what, just helping them. So hope they can motivated by this talk. Thank you so much.
So, son from Tamil Nadu, son of the soil, has come and established in Oman country. It's a great sir.
Thank you so much.
Thank you. I want to introduce my staff members to you.
Sure, ma'am. Sure.
First,
with pleasure.
Hi, sir.
Hello, ma'am.
Very
Good
nice
afternoon.
presentation, sir. And also very
Thank you, ma'am.
informative, sir. Thanks a lot,
Thank
sir.
you. Thank you so much.
She is Dr. M. Mumtaz, Assistant Professor of Zoology, the second man in the
Okay.
department.
Okay.
Our department is like one of the families, sir.
Okay.
We all sit
I will-
together and discuss and do everything.
I'm very curious to visit, ma'am. Next, during my visit to India, I will be
Okay,
coming and see what kind of
sure.
June and July, most probably in my vacation. So
Okay, sir, we'll invite you, sir.
thank you so much.
Our next personality is Dr. Rajapriya. She's going to deliver the vote of thanks, and she's the
Okay.
third man in the department.
Hello,
Madam.
How are you?
Convey the.
Madam, you can unmute yourself.
Ma'am, unmute பண்ணிக்கங்க.
Okay. Good morning. A warm and grateful morning to everyone gathered here. It has been such an honor to be a part of this wonderful event. It's my privilege to propose a vote of thanks who worked hard to make this international webinar grand finale. On behalf of organizing committee, I would like to extend my heartfelt gratitude to our esteemed guest, Dr.Senthil sir, who spared time from his busiest schedule to grace the occasion. Today, we had an opportunity to hear your thoughts, and this will surely be going on to encourage us in our future. Your presentation has enlightened our minds and have shown us a new path. My gratitude to you, sir, for gracing the occasion and sharing your research today. A special thanks to our college secretary and correspondent, Dr. S. Anishekar IAS, District Collector, Madurai, who shows keen interest in the development of the college. I thank you, sir. Without captain, the ship will not reach the destination. Likewise, our respected principal, Dr. A. Muhammad Aslam, sir, for being the catalyst that stimulated us to do our best and standing as pillars of strength. Thank you, sir, for your constant support. An event like this cannot happen overnight. The wheels start rolling weeks ago. It took us planning and a bird's eye for details. We have been fortunate enough to be backed by Dr. K. Shamim Rani, Organizing Secretary. I'm immensely thankful to you, ma'am, for your efforts towards conducting this international webinar. With a deep sense of appreciation, special thanks to our conveners, Dr. A. Mumtaz ma'am, Dr. M.M. Abdul Qadir Moidin sir, and M. Sikandar Bacha sir for their rock solid support and encouragement. I would like to thank Dr. M. Sheikh Muhammad Nias for reciting the verse from Holy Quran. Our sincere gratitude goes to all faculty members from my college and outside the college, technical, administrative staffs, and well-wishers for your supporting efforts. I would like to thank the people who work behind the scene to make this event success. Last but not least, on behalf of organizing committee, let me thank all students and all participants to be disciplined and honored. Finally, I would like to thank each and every one of you for being here and making this international webinar a wonderful and successful. Thank you. It's my bound duty to thank Professor Dr. Srinivasan for his time supporting in telecasting the webinar in platform and YouTube. Thank you, sir.
Srinivasan sir, எங்க பசங்களை எல்லாத்தையும் செந்தில் சார் காட்டணும். அந்த மாதிரி பண்ணுங்க. Srinivasan sir,
என்ன மேம்?
கொஞ்சம் ஸ்டூடெண்ட்ஸ் லேப்ல இருக்காங்க. கொஞ்சம் செந்தில் சார் காட்ட முடியுமா? ஃபீட்பேக் வந்துச்சு போடு. காட்ட முடியாதா சார்?
So your department, research department also, ma'am?
Senthil sir.
Yes, ma'am. Solunga.
Next, I'd like to introduce the third man in the department, Dr. M. Abdul Qadir Moideen.
Hello, sir. How are you?
Hi, sir. Good morning.
Hello, sir. Nice to meet
He
you
is
all,
Abdul
sir.
Qadir Moideen.
Okay.
He is assistant professor of zoology. He also worked in aquaculture, sir, from Dammam.
Okay, great.
So we are all glad to meet you in the month of
Thank you.
June or July.
Sure, ma'am. Sure, I'll visit you.
So thanks of you, sir. Convey our regards to your family members
Sure.
and your universities and your
Thank you.
head of the department, everyone.
Sure.
So thanks of you, sir. Thank you. Have a nice day, sir. Thank you.
I too had very good
We'll
experience.
talk later.
Yeah, sure. Thank you so much.
Thank you.
Hello, sir. Thank you, ma'am. Can I leave now?